Hi all, Dr. Clark here to talk about photosystem 1 and 2, or if you do it in the order they appear, 2 and 1. Okay, we'll get to this and I'll discuss a little bit about both these systems and go into detail about the construction of at least photosystem 2. Now, this is a diagram, a model of Photosystem II's P680 site. And so, we'll talk more about what the P680 site means. It means photon 680 or 680 nanometers is the maximum that the photon wavelength that this antenna complex can pick up. Okay, so remember when we dropped off, we were talking about antenna complexes, how they pick up photons, and through resonant energy, excite electrons. So this is a diagram of the P680 site in Photosystem II. And we'll come back to this in a second. Okay. So again, we left off, we were talking about how photosystems work, or how photosynthesis works in general. And we discussed that a photon comes in to the antenna complex. So remember, the antenna complex is inside the thylakoid membrane. Photon comes in, and it has some resonant energy. So this energy is captured by a single chlorophyll or single accessory pigment. Some pigment molecule picks it up, and then through resonant energy, it moves through the chlorophyll molecules to the reaction center. There's going to be a chlorophyll molecule in the reaction center that is holding on to an electron, okay, fairly loosely. Okay? And as that energy, that resonant energy, goes through the chlorophyll molecules, it eventually makes it to the reaction center and excites this electron. This electron then moves to an electron acceptor, and then that electron acceptor can move down what we call the electron transport chain. A new electron will come in to the center, and that comes in via the splitting of a water molecule, which is also a very interesting aspect. Okay? So let's look at this a little bit more detail. So again, in light-dependent reactions, that's what we're talking about. So we're talking about reactions that occur when photons are present. There are two systems, Photosystem II, and this is where a photon is captured. It releases an excited electron. That electron is picked up, moves to the electron transport system. That electron transport system produces ATP. A water molecule is split to replace that electron that was removed. And it's, on and on and on, as long as there's photons to be captured. Photosystem 1, okay, you get that electron from photosystem 2 that went through the electron transport chain. Okay, you get that in the reaction center of photosystem 1. Okay. Photosystem 1's reaction center, or antenna complex, is called P700. Okay. So photosystem 2 is P680, photosystem 1 is P700, and that refers to the maximum wavelength that is accepted in each one of those systems. Okay. So photosystem 1, that electron is excited by another photon, and it moves to the electron transport chain again. But this time, instead of producing ATP, NADPH is formed. Remember when we were talking about NADPH, we were talking about it being an electron carrier. Well, that's where this electron goes, is it gets attached to NADP to form NADPH, and then NADPH can carry it somewhere else. So again, the electron that's lost from photosystem 1 comes from photosystem 2. The electron that's lost from photosystem 2 comes from the splitting of water. Okay, so hopefully that makes a little bit of sense. Now we're going to go through each one of these in even more detail in just a little bit. Okay. So here's basically a diagram of exactly what I was just talking about. So photon comes in, resonant energy, excites the electron on the P680 site. The electron moves to an electron carrier, we'll talk about them in a little bit, and then moves down the electron transport system, okay, which is just a protein that allows for a proton gradient to form. So what happens is you have a protein embedded in the thylakoid membrane, and as an electron moves through that protein, a proton or a hydrogen ion moves through that gradient. Okay? And as it moves through that gradient, you get this buildup of protons on one side of the membrane. Well, through diffusion properties, those protons want to move back. Okay? They want to be at equilibrium. Okay? So you get a bunch of protons on one side, not very many on the other side. They want to move back. The only way they can move back is through a protein called ATP synthase. It's an enzyme. And those protons will move back through, so they'll come back through the membrane. And in doing so, ATP is formed. But that electron keeps sliding down the electron transport system, and it ends up at the P700 site. Photon comes in, excites it again, it's picked up again, it moves up, down the electron transport system again, or electron transport chain again, and instead of forming ATP and having a proton gradient, that electron is picked up by NADP to form NADPH. Okay? We'll keep going through this. It'll eventually sink in and make sense to the majority of you. I highly suggest if you're trying to learn this, the best thing to do is start to draw it out. So if you draw out where the electron's going, how it travels, what's the products, and whatnot, you'll eventually get it all down, and you'll commit it to memory. And then if you learn and look at questions and things like that, you'll eventually move from knowledge to being able to actually master photosystems. So plants can produce ATP and NADPH. Remember, ATP is the currency of the cell, so it's energy. NADPH is an electron carrier. And they do this by what we call non-cyclic photophosphorylation. Giant word. Okay, this would be, well, it's not possible, I don't think, to get that many letters on Scrabble, but that would be a heck of a point. A heck of a word to have. So let's start with non-cyclic. Most of you know what this means. It means it's not a cycle. So it's not a circular cycle. Okay? So this is a linear process. Water is broken down. The electron leaves the system. The photo portion refers to the photon. And then the phosphorylation refers to binding a phosphate to ADP to form ATP. So non-cyclic photophosphorylation. So again, these excited electrons will flow through both photosystems, photosystem 2, photosystem 1, and it ends up in the carrier molecule NADPH. Then the ATP molecules and the NADPH molecules can be utilized in what we call the Calvin cycle, or the building of carbohydrates. And so both those products, the product from photosystem 2 is ATP, the product from photosystem 1 is NADPH. Both those products are utilized in the Calvin cycle. And we'll get to the Calvin cycle next lecture. All right, so let's go into a little more detail in Photosystem II. Okay, so Photosystem II, this is where we were talking about biochemical pathways before, and I said that there's going to be some situations where you're going to have lots of proteins that are embedded in the membranes and have, in some cases have a structural role, and in other cases they might have a role in help breaking down different things, contributing electrons, these kind of things. So Photosystem II, its reaction center consists of more than 10 transmembrane proteins. And the reason why I don't give you a hard number here is it really depends on the plant species. Okay, different plants will have different numbers. Their photosystems are a little bit different. Okay, but they have a lot of transmembrane proteins in these reaction centers. That reaction center, where that free electron is held, is going to be surrounded by an antenna complex. Okay? And again, we already talked about the antenna complex. It's kind of like this, dish that houses pigments, pigment molecules, chlorophyll A, chlorophyll B, carotenoids, xenophils. These are all going to be sitting in this dish waiting to capture photons and then funnel that energy to the reaction center. That reaction center houses and holds an excited electron and will donate it to the electron acceptor. We'll talk about those electron acceptors in a little bit. Now here's one of the key points and one of the most interesting pieces of photosystems and photosynthesis for that matter, is after that electron leaves, right, and it takes 2 electrons for a carrier molecule. So this has to actually happen twice. But after that electron leaves, water is split. So you might think, wait a second, we know how to split water and gain energy from it? Yep, we do. And what is the waste product? Oxygen. So you're splitting water. The proton, the hydrogen ion, is moved through the membrane. Oxygen is left as a waste product, and a free electron is there. So yes, there are people working on this. Yes, there are individuals out there, engineers, that are trying to design hydro cars and things like that. Okay? This technology has been around for a long time. Plant biologists have known about Photosystem II for probably 70 years, if not 100 years. And they've known about this water splitting capability, and they know about the enzyme, one enzyme that can split water and do this. So that technology is out there, and I wouldn't be surprised in the individuals who are watching this in your lifetime that we see hydrocars or hydromotors that are running purely off of water, because we are moving in that direction. We're moving in renewable resource direction. There are countries that are over 50%, over 75% in some countries of the energy expended is renewable resources. And so with more technology, with more advancements, you're going to start to see some of these other things that we steal from plants. I mean, to tell you the truth, our whole solar panel is basically a photosystem. I mean, that's exactly how it is. I mean, that's where we got the idea to making photocells and things like that. It really came from photosynthesis. So will the splitting of water. Okay? So very interesting piece. I'm not going to go into a lot of details. If you're really interested in water splitting, there are lots of papers out there, and I can send you some. Just send me an email, and I can send it to you. All right, so that electron is going to move to the electron transport system, right? And then it moves down the electron transport system. And as it's moving down the electron transport system, that electron moves through proteins that are embedded in the thylakoid membrane. As it moves through a protein, a hydrogen ion can be pumped across from the stoma into the thylakoid space. Okay, so you're moving protons from the stroma into the thylakoid space, and you can get this proton gradient. So again, I think I mentioned this a long time ago, like chemistry. When I say hydrogen ion and I say proton, they mean the same thing. Okay? A hydrogen ion is a proton, okay, because it's just a proton. And so you get this proton gradient or hydrogen ion gradient. And like I said before, they want to come back across that membrane, across that thylakoid membrane. The electron at the end of the electron transport system is donated to Photosystem I, okay, to the P700 site, and Photosystem I. So Photosystem I is very similar to Photosystem II. Okay. A few more proteins to build up that reaction center. The antenna complex, same kind of thing though. It's still there to capture photons. It's working in the same manner. Okay. Now, in some plants, you might have, in your P680 site, you might have more chlorophyll A, and then in your P6 or P700 site, you might have more chlorophyll B or more carotenoids or whatnot. That can differ, but the general purpose, the general antenna complex is pretty much the same. But now, when that electron is excited due to resonant energy, due to the photon being captured, that electron is excited, it's donated, and moves through the electron transport system or chain, right? And then it reduces NADP into NADPH. Now remember, when something becomes reduced in this form, when we're talking chemistry, when something becomes reduced, it means it gains an electron. And in this case, it gains an electron and a proton, hydrogen. If something is oxidized, it means it loses an electron. So just keep that in mind, because we're going to talk about reduction and oxidation quite a bit as we move forward. So again, by having protons being captured and bound together in the form to form like NADPH, okay, you're also adding to that proton gradient. You're adding to what we call chemiosmosis. So you're adding to protons being on one side of the membrane and very few on the other side, and they want to get back across, right? And want in loose terms. It's just a diffusion mechanism where they're trying to move into an area that's less dense with the same molecule. All right, so let's check it out again, kind of in picture form. So you have water, okay, that is being split enzymatically, meaning there is an enzyme that can split water into oxygen and hydrogen. Okay? The oxygens will bind together to form like breathable oxygen, okay? And that will be released out of the system because oxygen is poisonous to plants. And then there will be a bunch of protons. Now, that electron is going to be donated to the reaction center from that splitting of water. Photon comes in, excites it, the electron moves through the protein, okay, and is donated back to the P700 site. That movement adds more protons, okay, in the thylakoid space. The electron then moves again through, and instead of forming ATP, which will be formed due to this process, see all these protons down here? It's not showing you ATP synthase. It's not showing you an ATP protein. That might be over here in the system. Okay, but that's where all these protons are going to go through that system. But that electron is going to be donated to NADP, to form NADPH. This is a misprint. There should be an H at the end of that. Okay, so it should say NADPH. All right, so let's look at ATP synthase. Okay, so that result of pumping protons, okay, gets a large concentration of protons, hydrogen ions, in the thylakoid space. Now, like all membranes that we've talked about thus far, things that have charges and things that are polar cannot move across the membrane. Okay? And that's due to the fatty acids. So you get all these protons on one side. They're trying to get back across, but they can't. Okay? The only way they can get across is if they go through a protein channel. Okay? And that protein channel that they're going to go through is called ATP synthase. Okay, so you know it's an enzyme, and based on synthase, you know that it's going to synthesize ATP. And it does so by phosphorylating ADP to form ATP. And the energy to bind another phosphate group to ADP comes from those protons being flushed back in to the stroma. We call this chemiosmosis. Okay? So there you get osmosis, so movement of things, across the gradient from a high concentration to a low concentration. And in this case, the chemi has to be like chemical movement. So here you can see it. Again, water split, hydrogen ions, bunch of them, okay? Bunch of them being pumped across as electrons move through the proteins, okay? And then they get back into the stroma, and they do so by going through ATP synthase to form ATP from ADP and inorganic phosphate. It's not shown here, but you'll have inorganic phosphates in the stroma also, and ATP will be formed. Okay? All right, so let's watch a little video demonstrating this. It does a much better job than the diagrams. It does better than my hand movements and my words. This is an excellent video that's produced by North Dakota State University and shows exactly what's going on.
The process of photosynthesis produces ATP from ADP and Pi by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: Photosystem II, cytochrome B6F, Photosystem I, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and Pi, which combine to form ATP. Photosynthesis occurs in only one cellular organelle, the chloroplast. Chloroplasts are found in all green plants, some algae, and cyanobacteria. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in Photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. One photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from Photosystem II are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the Photosystem I complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP and molecular oxygen are the final vital products of photosynthesis.
Okay, so I just want to reiterate a couple things. First, again, I disagree with the video. Oxygen is not a product. Oxygen is a waste product of photosynthesis. Secondly, the video just talks about pigment molecules being chlorophyll. And I know it's simplistic and the video is not very long, but there's other pigment molecules, and I want to make sure that you know the antenna complex is not just made out of chlorophyll molecules. It also consists of carotenoids, xenophils, other accessory pigments that are important for picking up photons that chlorophyll molecules cannot. The other thing that the video talks about that we're going to talk a lot more about is this connection between photosynthesis and cellular respiration. So the video says multiple times, just like in the mitochondria, just like in the mitochondria, just like in the mitochondria. Okay? That's because when we start looking at the electron transport system in the mitochondria and how material is broken down, the process is really similar in how things are formed, which gives us a lot of clues about the evolution of both these systems. Okay, so next time we're going to talk about the Calvin cycle. So now we've talked and we figured out how does ATP get formed in Photosystem II, how does NADPH get formed in Photosystem I. Now we're going to take those 2 products and we're going to utilize them in the light-independent reaction or the Calvin cycle. Okay, so next time we're going to do that.